Bonjour à tous, je pense qu'on peut commencer. Je vais faire une petite introduction en français, mon français. Et en tout cas, j'ai le plaisir de vous présenter Alain Herre. Il est un chercheur qui travaille à l'Institut Smithsonian de recherche tropicale au Panama, aussi une affiliation avec l'Université McGill. Donc, il a fait beaucoup de travail depuis longtemps au Panama, surtout dans l'île de Barro Colorado. Ça, c'est un laboratoire naturel de recherche depuis longtemps, je pense que 100 ans, 100 years. 100 ans, donc beaucoup de recherches, c'est très connu. Alan aussi, il a fait beaucoup de contributions à la science. Il a publié beaucoup d'articles dans quelques journaux obscurs et pas très connus, mais ça arrive. Donc, l'aspect théorique, l'aspect théorique aussi, la partie empirique. Donc, bon, avec plaisir, je...
Thank you all for coming. Today, this is meant as a very informal talk. I realize most of you do not have English as your first language, and I apologize that my second best language is Spanish and not French. So I'll speak slowly, but if you have questions, please ask. Today is in fact extremely informal and perhaps a little bit different than what you might normally be accustomed to in terms of a talk, because as I have talked to students coming here and as I have seen that the different lines of inquiry that people study here, I would like to give you guys the option of hearing at least some of the specifics of what I will talk about. So broadly speaking, I am very interested in how very small things, be they fig wasp or be they mycorrhizal fungi or be they endophytic fungi that are in leaves, affect much larger things, plants, trees, communities, animals that eat fruit. And so my broad interest is how tiny things do or do not form associations with the larger things. And when they do that, what is the outcome? Is it a parasitism? Is it a mutualism? Is it a benefit for one or both? And what are the mechanisms? Certainly, my original intent was to give a broad introduction to these kinds of questions and to bring up broad kinds of issues in the study of mutualism, and then more or less leave it open to you as an audience to tell me what aspects you're more interested in. And your options are figs and pollinating wasps. If you like figs and pollinating wasp, the kinds of options there, in fact, the study questions are, what are the levels of coevolution between these? What are the mechanisms of interaction? And what controls whether or not one is beneficial or parasitic on the other one? In endophytic fungi, which live in leaves, the topics there are associations of the fungi with the hosts, how specific are particular fungi to particular hosts, and what are the mechanisms of effect that the fungi have on the host, and to some extent, vice versa. With mycorrhizae, the general questions that I and my collaborators are currently trying to understand is, what role do mycorrhizae have inside of tropical forests, particularly tropical forests, in terms of modulating how pathogens affect hosts? This is important, we think, because most evidence that exists to now from these areas, this is Barrow, Colorado Island, suggests that the diversity of the forest and the relative abundance of the most common individuals is influenced by their, or at least primarily by their interactions with soil communities. This is not to say that the soil microbial communities are the only factor that influence the trees, but it's one. And certainly up to now, most people have considered the effect of pathogens on hosts as the primary driver. To give you some background of what I mean by diversity, this piece of land that you can see with this boat conveniently for scale has got about as many tree species as all of North America, Canada, US included. So one of the things we're very interested in is diversity. And just to give you another sense of diversity, huge number of tree species here, that kind of diversity in fungi can be inside of a single leaf, just in terms of species present. I can talk about that also. But just to return as part of the general introduction, I'm very interested in how small things affect bigger things, how the associations form, and how each partner is influenced by that interaction. As you can tell, there's a tremendous amount of overlap and common ground with Juan Carlos. And so, at any rate, part one. I am mostly going to be talking about mutualisms, which generally is thought to mean both of the partners, both of the interactors gain some kind of tangible, usually measurable benefit from the interaction. My motivating questions, at least for this talk, are how stable are mutualisms, and in what senses are they stable? Are they phylogenetically stable? So in other words, are the genetic or the species associations over time stable? Are they ecologically stable from place to place to place? Are they functionally stable? So the effects and the interactions, and what kind of factors can contribute to the stability or lack of stability of these different sorts? Okay, so I just want to mention as I go into it, there are a lot of very instructive systems of interactions of mutualisms, and almost every single one of these is characterized by a host acquiring its symbionts from the environment, as opposed to a host acquiring all of its symbionts from its mother or parents. It acquires them from the parents. It's called vertical transmission, it's shorthand for it. And in that case, it's very obvious that the symbiont, which is passed from a parent to an offspring, really wants, and I use the word in quotes, but really wants to have the offspring that it enters be successful, because its success is very tightly linked to the success of the offspring. However, in the case of symbionts, be they microbes, be they viruses, be they fungi, be they bacteria, be they insects, whatever, if they are acquired from the environment around any individual organism, potential host, has got an extraordinarily wide, diverse set of possibilities of small things that might form an interaction with it. Some of those could be incredibly beneficial. Some of them could be lethal pathogens. So the point here is that if you compare broadly a vertically transmitted symbiont with a horizontally transmitted symbiont, when picked up from the environment, you think beforehand, a priori, that the horizontal cases are the ones that are actually more interesting because it is less obvious that those should be mutually beneficial. And so in the horizontal transmission cases, which all of these things represent, what is it that ensures that the interactions that result are actually beneficial, if they do ensure it at all? But just to walk down these, nitrogen-fixing bacteria in roots of most legumes, mycorrhizal fungi that allow most host plants to acquire phosphorus, endophytic fungi, which are in the leaves or in the branches, in fact, also in the roots of plants. But I will focus on foliar endophytic fungi, fungi in leaves. Yuccas and the pollinating moths, Glitchidium and the moths that pollinate those. This is a fairly recently discovered pollination mutualism, which is extraordinarily diverse across the world. Corals, the algae which inhabit them. Generally speaking, the algae are photosynthesizing and providing some sort of nutrient supplement for the corals. Leaf-cutting ants in their fungal gardens. This is just a tip of an iceberg. The ants cut leaves, they feed it to a garden of fungus. Fungus feeds the ant. There are a lot of other things associated with that as well. Figs and pollinating wasp, which is what I have done most of my work on, sponges and their microbiomes. Simply put, any environment that you look at in Earth, whether it's terrestrial or marine, many of these, in fact, most of these, have at their base some kind of mutualism, which is helping to structure entire communities. And so each one of these is really interesting and very important in its own right. If you look across them, then you can begin to say, are there things like common patterns? I do not know the answer to that. I simply put those up so that you start thinking about it. And so here's the question. Are there any general principles that apply across those, or is it simply a lot of different, extremely interesting special cases? How general, how special? Are we even asking the right questions? But in any case, we need information. I think you guys have heard quite a bit of it from this gentleman right here. But fundamentally, this is a comparative undertaking to see what is similar, what is different across these. So, to get back to one of the first points, how stable are these? Okay. At some kind of taxonomic scale, these are incredibly stable. There's no question about it. You can go back in fossil records, you can find sponges have been associated with their microbiota for over 600 million years, plants with mycorrhizae, 400 million years, plants with foliar endophytes, again, 400 million years. When plants first came out of marine systems, they had mycorrhizae, they had endophytes. Corals and their algae, at least 200 million years. Figs and fig wasp. This broad system I'll describe in detail, depending on how much detail you want. They've been extant since the time of dinosaurs, since the time when apparently most continents were either in the south in Gondwana or in Laurasia. I personally think that figs started in the southern continent of Gondwana. A group of old world fig biologists think or want to think that it was in Laurasia. It doesn't really matter, it's old. Atine ants and their fungi, 55 million years ago. Legumes, rhizobia, about 55 million years ago. Aphids with their bacteria, I had thought it was 30 million years ago, but about 3 months ago, I learned that that figure has pushed back 100 million years. So these kinds of interactions, these general forms of interactions, all evidence suggests that they are very old. Okay, so at some level, yes, they're very stable. On the other hand, there's also very ample evidence that the mutualisms break down and change almost constantly at both a broad taxon level and at a species level, particularly at a species level. We see switching of hosts. We see gain or loss of a kind of benefit or cost of an interaction. There's functional switching from mutualism to parasitism. And so if you ask the question, are they stable? I'm going to say, I'm going to present you guys evidence that on the one hand, if you ask it at a very broad scale, yes, they're very stable. But you also can find scales at which they're very fluid, very flexible, very dynamic. And so, let's see. So I'm going to give you some examples of ecological and evolutionary instability, phylogenetic incongruence, breakdowns between the symbiont and the host, and the degree of their species specificity and their functional relationships. If at any point anyone has a question or a comment, please question or comment. Okay, so I'm going to start off with host tree species. These are leguminous trees in the tropics with their rhizobia, with the nitrogen-fixing bacteria. All right. What you're looking at is data from a study that was conducted in Panama. And here, the people had a large area of forest, some of which had been cut down at different ages. So they had pieces of forest that were very old, pieces of forest that had been in the process of regrowing, had been regrowing about 75 years, another piece that was about 30 years, and then some which was just beginning to regrow. So the idea is simply you have a big plot of land which has got some of it that has been cut at different ages, and the forest has regenerated in those areas. What they found was this. If you're interested in the total proportion of the roots that have got nitrogen-fixing bacteria, which are, you can tell they're there because of the formation of something called a nodule, they saw this very interesting pattern. The very youngest forest had very low proportions of these, but then within 5 to 10 years, the regrowing forest was filled with these nodules. This means that the roots had high densities of nitrogen-fixing bacteria on their roots. They did not look at the species of the rhizobia, of the nitrogen-fixing bacteria, simply presence of the structures. The different colored lines are different host species of plants. What you can see is that if you go across time, there are huge differences in the proportion of the roots which are carrying the interaction with these microbes. And if you ask a question of, is this stable? Well, it kind of depends whether you're looking at a piece of forest that's about 75 to 80 years old, and here you would go, well, oh no, these trees don't even have it. Or if you're looking at one which is 70, or I mean 30, whatever, what you're seeing is variation is a piece of the whole problem, the whole question. There is not a really straightforward answer here. Is it stable? Is it not stable? It appears as though during the course of their lifetime, trees are gaining and losing these relationships. Studies from other parts of Panama suggest that in more intact forests, the level of infection is actually quite low. But if you look inside of tree fall gaps where trees are rapidly growing, it's very high. And if you look in areas where that's along a shoreline with lots of light, lots of water, it's also very high. But the point here, I think, which is worth making, is that it changes. It changes over the lifespan, probably, of a single tree. These are different trees, albeit of the same species, here from here. So what you would really like to do here is start with a set of trees, look over their lifetime, and see how it changes. But this immediately says there is some very fundamental degree of shift of the relationships of the hosts with with their microbial symbionts, their mutualists. Okay, now what I've just said, it would be good to look at how individuals change over time. This is a snapshot with trees of different ages. I'm about to show you a case where you can look over time. And in this study, which is with mycorrhizae, my collaborators here were Dr. Rebecca Husband and Dr. Anhum Om. Dr. Om is an expert in mycorrhizal fungal morphologies. You can see some of the very pretty fungal spores here. The problem with these things, as beautiful as they are, is that once they form an association with a plant root, it's very difficult to identify these things to species. And so the technique which Dr. Husband was in the lab that pioneered it is to use basically metagenomics. At the time we did the study, it was Sanger sequencing with very tedious cloning. But that gives this following kind of data. This is a study done about 20 years ago. We started in Panama in the beginning of a rainy season when lots of seeds had been dropped of a common species, Tetragastris, and there was a seedling carpet. We tagged all the seedlings, and then each year we would come back and sample from the same cohort. So it's still not the perfect sampling, but at least we are sampling of the same group of seedlings. What you're looking at is color coding for the species ID of the most common mycorrhizae with, in this case, 3-month old seedlings, 98, 1-year-old seedlings. So these are the ones that survived 1 year, and then to 2 years. And then on the second year, we also collected saplings in the general area. So much older plants of the same species. So it's still not the perfect data that we want, but it begins to move in that direction. But what you can see is if you ask a question like, is there a really high degree of specificity between the mycorrhiza and the host? You sample here, you go, no. You sample here, go, well, this one's really dominant. You sample here, you go, oh no, and that one's dominant. You sample here and you go, well, yeah, this one's very dominant. But who is dominant is changing over time. And we interpret this as being something that's akin to succession inside of hosts. So when you sample a host, how old it is, its physiological condition, its ecological setting, all of those kinds of things appear to affect the answer of, is it specific? Is it stable? So if someone says, no, it's not stable, no, if they're right. If someone says it is stable, well, they're right too. But it really kind of depends on the way that you sample these things. At least this is our current take-home from these, take-home message from these studies. So what do these things do? This is an example from a study we conducted where we took Theobroma cacao seedlings. These are the seedlings of the plant species that produces chocolate. I think everybody likes chocolate. Well, most people like chocolate. And they were either grown in soil that had mycorrhizae or soil that did not have mycorrhizae. So this is trying to get at function. Then we would challenge the leaves with a pathogen. In this case, it's Phytophthora, no mycete. And in the mycorrhizal-free soil, we added a lot of phosphorus. At least previously, many people thought that the function of mycorrhizae was to give the host phosphorus, was to make phosphorus more accessible to hosts. Well, we did that. But what you can see is that the plants that were challenged with the pathogen had much less negative results or reaction to the pathogen if they already had a mycorrhizal association in their roots. Keep in mind that the mycorrhizae are connecting with the roots, not with the leaves. So this is a distal effect. And so this is trying to get at what are these fungi doing with the hosts to break quickly to a conclusion here. No, I will not break quickly to a conclusion. Instead of that, I'm going to jump to the foliar endophytic fungi. So rather than being in the roots, these are fungi that are in the leaves of essentially any healthy plant that you see. This is a picture taken by my Panamanian collaborator, Luis Carlos Mejia. And what he did was clear the leaf tissue. You can still see the cells and stain the fungi, the endophytic fungi that are living literally inside of the tissue in the interstitial spaces for the most part between cells. So this is conceptually what almost all plants' leaves look like. You take a leaf, you take a slice of the leaf, and then you take tiny slices of it, you put it on agar, you grow out the fungi that are living inside of a healthy leaf that shows no evidence of any kind of pathogen damage. And what you find, these are codes for different species of fungi. You find lots of different species of fungi living inside of healthy leaves. What do they do? I will try to tie this back to the mycorrhizae in just a moment. One thing that they do, you could think of this as being that, this one as being that, etc. They produce antibiotics and chemicals which interact with other microorganisms that either are or that might be inhabiting the leaves. So inside of a beautifully healthy leaf, you will have all of these kinds of things growing in it, or at least alive in it. They will be producing these chemicals. These are usually chemicals that the plant itself does not produce. So they are something in addition to what the plant could produce by itself. And they have inhibitory effects on bacteria, other fungi, etc. So it took us a great deal of time, but broadly, this is what the life cycle of these things is. The plant, when it flushes new leaves, those leaves, to a first order approximation, do not have any fungi inside of them. They are flushed into the world, basically free of these micro communities. With time, the leaves will have fungal spores land on them. The spores germinate and will work their way into the tissue. So they acquire their fungal communities from the outside habitat. Over time, the density and the diversity of those fungi starts to go up. And then over time, what we find is differential proliferation of some of those fungi. So there is a process which is analogous in the leaves to what we saw in the roots with the mycorrhizal fungi. At some point, the leaves die, they dehisce, they drop, and then very rapidly, the fungi which are already established inside of the leaf take over. They break down the cell tissue of the leaf, and they release more spores, and they start the process over again. So that's the rough life cycle of these guys. One of the things that's interesting about the endophytic fungi is they're often very closely related to pathogens, things that are known plant pathogens. One of the fungi that we worked on, Calototricum tropicale, had been previously lumped with a lot of different Calototricum as a very polymorphic species. But detailed genetics suggest that, in fact, this polymorphic species is a cluster of more well-defined species, which themselves are either pathogens or symbiotic, potentially even beneficial, and mutualists. So whether a fungus or a lineage of fungi are a pathogen or not, is actually quite labile evolutionarily. This is work from colleagues who are working in Mexico. But the most important point here is just look at the number of transitions between black, which here is, I think, a pathogen, to brown, which is an asymptomatic endophyte. It's not as though there's a group of pathogens and a group of endophytes. They're switching back and forth. In the foliar endophytes, my colleague Luis Mejia spent about a year of his life to be able to do this. We can produce plants that do not have endophytes, and you can do that simply by growing them either with their leaves kept dry. Leaves that are kept dry, the spores that land do not germinate and do not work their way in, or you could keep them in a filtered air environment, but as long as you keep spores from access to them or from germinating, you can produce endophyte-free tissue. Or you can produce tissue, which is sort of the normal state of most plants, and you can control which endophyte species actually go into a host. Why is this interesting? Remember the picture that I showed you of the mycorrhizae? It's a similar kind of study with endophytes. We can control whether this leaf on the plant does or does not have endophytes, and endophytes of our choosing. We can say, well, is it a common endophyte in healthy hosts? Yes. Well, if it's a common endophyte in healthy hosts, that suggests that at a minimum, it doesn't hurt the host usually. So it's a good candidate for something which, if nothing else, doesn't hurt it, and it might actually be beneficial. So based on that criteria, we basically made a big soup of the most common endophytes, inoculated a host. Some of the leaves of the same individual plant did not get inoculated. And then we did the similar experiment where we challenged it with a pathogen. What we found was that when you don't have the endophyte in the host leaf, pathogen damage is much more extensive. So by having these things, the host is gaining some level of resistance against pathogens. Are there any questions at this juncture? Yes. That took us a couple of years to work out. Yes, it's extremely important. The endophytes, to a first order approximation, can provide some protection within a couple of days. So you inoculate, then 2 or 3 days later, you treat with the pathogen. Generally speaking, the longer that you have the endophytes in the host, the greater the resistance is. We haven't done the limiting case, which your question implies, about co-infection at the same time. That's on the research docket. But yes, but everything that we have worked on exactly that up to this point suggests that very rapidly, within 24 hours, the plant that is treated with endophytes is beginning to upregulate parts of the innate immune system that it uses to basically protect itself. Our working hypothesis is that, at least in this case, with this endophyte in this plant, the primary benefit that this endophyte gives the host is that it is causing the host to go, oops, I'm being invaded. I'm going to upregulate defenses. There are other endophytes, other species, where we've got evidence that they also produce chemicals that the host would not normally produce. So you have an intrinsic effect to the host. You also have an extrinsic effect in terms of the chemical that they produce. But to continue on that point, we see the very first effect of the endophyte treatment being a downregulation of host defenses. We think the endophyte is saying, okay, turn defenses down, I'm going to come in. Once it establishes, then they go up. And so somehow the endophyte is acting like a pathogen in the sense of causing the host defensive systems to go down, but rather than go down a pathogenic route, it goes into a route where it is, after it establishes, it upregulates host defense, but does not cause at least obvious damage. But it does not, and by doing that, it gives the host more defense against other things. However, it also reduces photosynthetic capacity. It starts shifting nitrogen and carbon allocation. It affects water balance. It does a lot of things to the host, some of which clearly are not beneficial. And so our rough interpretation is that many of the microbes that are inside of the host system are inside of healthy hosts are actively manipulating the host's defense systems so that they can establish themselves and prevent, to some degree anyway, other things from coming in. But yes, timing is a huge amount of work. Yes?
By using plants and your Phytophthora as the assay, you're isolating endophytes that produce these antimicrobial, even anti-cancer drugs. Has any pharmaceutical look at your system or using these approaches?
Not as many as we would like them to do. And we would certainly try to encourage them to give us the kind of research budgets that someone like you could, if you want to.
Happy to help. That's part of the work.
So it is not ignored. It is not ignored, but in the same institute, there are other groups which are more interested in, let's go back to, oops, no, we're going forward to this. You isolate things, you cut this out, you go, what's in there? What follows from your question is that for any given plant host, we can say, here are the most common endophytes. To a broad first brush approach, most plant species will have most endophyte species present, but there are usually differences amongst them in which ones are most dominant. We think, we have evidence that the most common endophytes in any given host species will grow best in agar that is infused with chemicals from that host. So the ones that are not so good don't like that host as much. So we immediately have got some evidence that host chemistry is at least suggestive of which endophytes are going to be common. we now have the other piece, which is endophytes in a host will upregulate certain sets of host chemicals. We don't yet know if the chemicals that an endophyte upregulates are also the ones that it personally likes best. We suspect so, if this is in fact a beneficial mutualism. But yes, the door is open on precisely all of these things that you're suggesting, and we are beginning to get at it. It has taken us 10 years to go from assaying across different fungal species, different host plants to know what fungus are common, to being able to do these manipulations. But yes, it's exciting. Other questions? Okay. So, oops, I keep going the wrong way. Okay, I'm sorry, I keep going the wrong way. Okay, let's try to go the right way for a change. So here's a summary of what I've talked about so far. Essentially all plant tissues, even particularly or even perfectly healthy plant tissues. In fact, I would even go as far to say particularly healthy plant tissues are filled with live fungi, leaves, stems, roots, and different groups of fungi are generally associated with different types of host tissue, leaves, stems, roots. The presence and identities of these fungi influence many of the characteristics that we usually associate with a host. It affects their growth, their allocation. We've just finished an experiment where nitrogen pickup of hosts is influenced by whether or not they have endophytic fungi. People know that if you've got mycorrhizal fungi, it influences phosphorus. So response to and access to nutrients, chemical composition. I think people frequently say, oh yes, this host plant is typically or characteristically got these chemicals in it. What people do not know, or just beginning to recognize that they should know, is which of those chemicals are actually host generated and which ones are microbial generated. The genetic content and expression, we know that which cacao genes, because we had very nice genome information, by having endophytes in them, different parts of the host genome get either upregulated or downregulated, physiological, photosynthetic, again, defensive. All of those things are influenced by the microbiota, in this case, the fungal endophytes. It also influences defense, antipathogen, I haven't shown you, but also anti-herbivore properties of the host. A broad take-home is simply that people who are very interested in plant genetics or plant genetic expression that do not include endophytes that are normally part of a host are at the best are incomplete. And by that, I mean a lot of growth chamber or greenhouse experiments. In a study, which I will mention but not show you, I can show people afterwards if they're interested. We could control the composition of the endophytic fungal community in a host plant by altering the, or by varying the composition of the leaf frass, cascara, hojarasca. Okay, anyway, but the leaves that you put in the base of a pot, if you put dead leaves of a host which was healthy, so dead leaves from a healthy host, the seedlings that grow from that in that pot are more resistant to pathogens than ones that are grown in a pot without any leaves or with leaves from a different host plant. So if you guys are gardeners and you have got healthy plants, cherish your healthy plants, take their leaves and put them around the base of the seedlings and the newly planted seedlings of the same species and see what happens. You can run your own experiment. But I have talked now for at least 40 minutes. I've got a lot more slides, but I will save them and I can use those to answer questions that you might have. But this is an overview of the kind of work that we are interested in doing with endophytes and with foliar endophytes and with mycorrhizae. Thank you. Yes.
If you take some of your plants that you showed us, I assume that they were from the environment, from some forest somewhere, as you look at these endophytes. Can you take the same plants and then grow them in a greenhouse and then examine to see if you're getting the same type of endophytes? Do they produce the same molecules, or is there a dramatic change when they're put into cultivation? Would that have an impact? Or are we talking about natural plants in the forest and you sample them?
Both of you guys have asked questions which took us years to work out. This experiment was the first experiment that we did. We got a very strong result. We did this in a greenhouse. And so this leaf would have been painted with a spore solution of endophytes. This leaf would have been untouched. We tried to repeat this 2 months later, got no result. Tried to repeat it again, got no result. That was when we figured this part out, which was the spores are in the environment. If the leaf is wet, they will germinate and they will grow in. We conducted the first experiment. It was like a gift. We did it during dry season. It was so dry for so long. Even being dumb about this, it worked. And we didn't realize why it worked. And it worked because our control leaves were really endophyte free. And they were endophyte free because it had, by luck, been so dry. But if you are working in a greenhouse, you are going to have different kinds of aerial microplankton than if you are in a forest. If you are in a forest next to a conspecific, where a lot of leaves from that tree are in the leaf litter, you're going to have an effect that you would not have, or that will be slightly different than if you were 50 meters away. And so trying to realize the relative importance, or the existence, and then the relative importance of these kinds of factors has been a huge piece of our research. And again, it was last year that we finally put the pieces together that at a very fine scale, you can influence which fungi are common or not by making conspecific leaf tissue available to it. But yes, it's the system is so complex that it is easy to just say, oh, there's no sense to it, there's no pattern. But then slowly you begin to realize, oh, these are the factors that influence it. At least that's our current working hypothesis. Yes.
So you show us an example like in a tropical area, but I wonder like in more like temperate area where the diversity, like species the diversity is usually lower. Do we also have like such an important number of like fungal endophytes, or is it lower or much lower? Like for example, in conifers in Canada.
Conifers have a very different, okay, this is not my study. They have a very different group of fungal endophytes that are associated with them compared to deciduous trees. There is a conflict depending on whose study you believe. One says there is a strong temperate tropical gradient with a higher diversity in the tropics. That's what I tend to think personally is correct, if for no other reason than you've got so many different kinds of hosts in the tropical area that there's opportunity for more types of endophytic fungi to coexist, because some could be common on one host and then jump from one and survive someplace else. In this kind of area, you have many fewer host species as potential hosts or guests for these things to be guests. So my suspicion is, yes, there's a difference in overall diversity, but different studies have come to different conclusions, in part because they will go, I will use metagenomics and I will use a 99% cutoff, similarity cutoff between ITS sequences of these things. Or another one says 97. And so it is important to look at the details of the different studies and how they actually define a species. Regardless, it doesn't make any difference where you are. There's a lot of stuff. I mean, that's true. How different? That's more difficult. But there are a lot of things there. I mean, how useful is that? I mean, anyway, I'm sorry it couldn't be more useful than that, but that's the nature of the problem. Any other questions? Yeah.
Sorry, if there is any sort of coevolution between an undefined and a plant, is the plant at a certain stage able to recruit the undefined without the undefined sporulate its way through the plant? Like the plant will produce a specific compound to recruit those undefined.
Okay. I think a plant, my guess is that a plant cannot actively say, bring an endophyte to it. It has to wait and whatever lands on it is what lands on it. What it can do is produce chemicals that will promote some subset of the fungi that actually do land on it. That's our current thinking. In terms of putting that in coevolution, I suspect that plants which are more able to choose and promote beneficial fungi by producing chemicals, by the molecular crosstalk, those ones will generally be more successful because they generally will tend to be colonized by more beneficial fungi. On the other hand, plants get sick, they die. So I suspect there is some kind of coevolution. We aren't far enough, or I'll put it differently, Luis is not far enough in understanding the details of the genetic interactions to be able to say, in this pathway, this host responds to this fungus in this way, this one becomes more beneficial. We have run experiments where we've taken the beneficial fungus, we'll call it fungus A, and we've got a cousin or a distant relative of it, we'll call it fungus B. They have very similar signatures of effects on the host induction, but there are about half a dozen host defensive pathway genes which are very different in terms of how they get expressed between the beneficial one and the pathogen. That would be the raw material for coevolution to work on. But we don't yet know enough details about that other than to say they're different. So yes, I think that there are going to be broad coevolutionary patterns, but you always keep that in mind with the fact that there are also pathogens which are trying to break those codes and break into the system. And the broad phylogenetic data suggests that there's a lot of shifting back and forth between pathogenic and mutualistic outcomes. So there's a lot of stuff to do.
Okay. Thank you very much.
All right.